Next speaker is Andrew Kern from Dartmouth College. He's our youngest speaker and probably dealing with sort of the youngest contribution to the field, genomes, basically. And basically he's going to discuss something along the lines of what John's been talking about here, how to basically detect selection in whole genomes. The title of his talk is The Tortoise and the Hare, Finding the Targets of Selection in Genomes.
So yeah, first let me thank the organizers of this event for inviting me. It was a very gracious invitation, and hopefully I can live up to their expectations. John really gave you a great warm-up for this talk. He was talking about some of the theory behind the practical applications that I'll be talking about today. So I was asked to talk about genomes a little bit, and I wanted you guys to imagine at the start of this talk a world in which there was no Darwin. So imagine there was no Darwin, there was no evolutionary theory, but there were still genomes. What would you do when Craig Ventnor came around? What would you do with this information, right? CTAACCCTAACCC. Is this making sense to you, right? How do we actually understand genome sequences? Well, the bottom line is that evolutionary theory fills that gap. Because I'm kind of a rambunctious character, I like to poke fun at these guys, Kimura and Oda, and we've talked about them in their neutral model and their nearly neutral model quite a bit this morning. In 1974, these guys wrote a paper concerning some principles governing molecular evolution. In particular, they had 5 principles. The first one was the Zuckerlund and Pauling observation, proteins evolve in a clock-like kind of manner. The second one, which I'll be talking about quite a bit, is that when clocks do differ, they say the clocks differ due to functional constraint on molecules, okay? Certain molecules are more important to biology than others, right? So histones evolve slowly, say. Fibrinopeptides evolve rapidly. Their third principle was that the majority of substitutions are going to be conservative. This is kind of just like number 2 here. They had something about gene duplication, which we'll ignore for the time being. And their fifth principle was that purifying selection was common, whereas adaptive evolution, like the kind of evolution that John Wakely was talking about, is actually really rare. Like I said, I'm going to be concentrating on principle number 2 here. So principle number 2, the fact that clock-like variation depends on functional constraint, is actually the very shallow intellectual heart of comparative genomics. Okay? That is to say that when all of our friends at the sequencing centers go and they sequence 44 different vertebrates, this is the current state-of-the-art UCSC genome alignment, when they go and sequence all of these vertebrates, what are they hoping? Well, what they're hoping is that when they align them, they see stuff like this. That is, what we're looking at here is a shot of the UCSC genome browser, okay? And if we look at this track, this is conservation kind of going up and down in these peaks and valleys. In particular, what our friends, the comparative genomicists, are hoping is that this variation in conservation tells us something. It tells us, in particular, where the important parts of the genome are. So have no mistake, okay? Evolutionary theory is at the heart of modern genomics. We need this stuff. The bottom line, though, is that Kimura and Oda have this kind of very, what I consider a dour perspective of evolution, right? The majority of evolution in their mind was either neutral or under a purifying selection regime. That is, the majority of mutations are deleterious. Well, we know evolution is otherwise, right? And how do we know this? We know this because of casual observation of the natural world. Not all of us get outside very often. Some of us live in very cold climates, so we can turn on the Discovery Channel, right? And bear with me, what happens when you turn on the Discovery Channel? Well, you see amazing organisms like this. This is one of my favorite organisms. This is the leafy sea dragon, okay? It's a fish, right? But it looks like a piece of seaweed. Now, is this the result of small effective population size? Probably not. This is a leaf katydid, another great organism, right? I use this to teach my introductory biology class. It's a bug. It looks like a leaf. And it looks like a leaf down to the point that there's this fake leaf hole in it, okay? Now, who in this audience is going to stand up and tell me that that's the result of stochastic processes due to demography? I don't think any of you. So when we see these kind of things, this is the guy we're thinking about and the guy who we're celebrating today, Charles Darwin. And I would also like to say that this is someone's name who I haven't heard mentioned yet today. We should also be thinking about John Gillespie, who in the face of Kimura and Oda really championed the neo-Darwinian approach to molecular evolution. So here's the cartoon, right? If I want to turn some plain fish like this goldfish into something spectacular like this leafy sea dragon, well, what is selection going to do? Selection sees phenotypes, but really it's going to go and hammer on the genome. Okay? So this suggests that if we're interested in looking for interesting bits of genomes, if we're interested in looking for functional bits of genomes, we should be looking for the signal of selection. For this talk, there are really two kinds of selection I'll be talking about, negative selection or purifying selection. To illustrate this point, let's compare the anatomies of a chicken heart and a human heart. Okay? These are two organisms which have been diverging for 310 million years, very long period of evolutionary time. But amazingly, the anatomy is very similar. Okay? That is to say, at least from my perspective, both of these hearts have 4 chambers. Any chicken that was born with 3 chambers or 5 chambers, probably dead. Okay? So this is the Kimura and Oda worldview. The majority of new mutations are deleterious. Okay? Conversely, we can think about positive Darwinian, sometimes called positive Darwinian selection or adaptive evolution, adaptive selection, whereby we get kind of this amazing fit of organism to environment, okay, over and over. And this is 3 examples just from leaf katydids, having these incredible bite holes out of them, just astounding stuff. So this is the case I'm trying to make. In genomes, function equals selection. So what are our tools? How do we go and find selection in genomes? Well, the more familiar approach to this might be the comparative genomics worldview. So what we're going to do is we're going to align divergent species genomes, put them in some kind of multi-alignment, say, and what we're going to look for is we're going to look for different regions of conservation. So if we're interested in finding negative selection, like the majority of the comparative genomics world is interested in, what we should do is we should look for conserved regions of genomes. So in this case, I've highlighted a conserved amino acid residue, this lysine here, that's conserved from humans all the way down to zebrafish. This lysine happens to occur in a gene that's thought to have been positively selected in the human lineage, the FOXP2 gene. So if we're interested in going and finding positive selection, what are we looking for? Well, often what we're looking for is lineage-specific change. So if we look in this residue here, what we see is that from chimpanzee to this fish fugu, the puffer fish, everybody is a C, but humans have this lineage-specific change to an A. While that comparative genomic worldview is more familiar, it's not necessarily very good at testing hypotheses. So I like to point out to people that really sequence comparison is very good at generating hypotheses, but you're not really getting, phylogenetics currently doesn't often deal in mechanism. Population genetics, however, does deal in mechanism. So once we have hypotheses, we can often test them using population genetic data. In particular, the beast that I'd like to talk about is the site frequency spectrum. And what is the site frequency spectrum? Well, basically what it is it's the frequency of genetic variants at some locus, okay? And what's nice about the site frequency spectrum is it's exquisitely sensitive to things like selection. It's also sensitive to things like demography. We'll ignore that for the majority of this talk. So here's an example for the uninitiated. What is the site frequency spectrum? Well, it's the distribution of allele frequencies within a sample. You'll imagine that I sequenced 4 human individuals and 1 chimpanzee. And if I go from left to right, what I'm going to do is I'm going to find mutations. I'm pulling a little bait and switch here. What I'm going to be talking about is derived allele frequencies. So these are new mutations, the red mutations here. I'm going to go through this alignment and basically tally up the counts of each of the different frequencies. So at this first site, this G is at a frequency of 1 out of 4. I get 1 count over here. This A is at a frequency of 2 over 4. I get that count over there. And I go through this, and basically what I get is a histogram of frequencies. Sometimes the site frequency spectrum is a little confusing to people because of that, because it's the frequency of frequencies, right? It's a histogram of frequencies. Bear with me. I just want you guys to have a gestalt appreciation for what I'm talking about. What's very nice about the site frequency spectrum is that through the mathematical machinery of population genetics, we can have an expectation for what that site frequency spectrum should look like under different models of evolution. Okay? So if we were to say, to compare the neutral model, illustrated here in yellow, what we see is that the neutral site frequency spectrum has this kind of L shape to it, right? The majority of variants are rare, and there are decreasingly, higher frequency variants are less and less abundant. Conversely, under a model of negative selection, where new mutations are deleterious, the majority of frequencies in a population or in a sample are very, very rare. John was showing us some of this earlier. The third thing to compare this to is under a model of recurrent positive selection, where what we see is this tail of high frequency variants as new mutations are going through the population, eventually sweeping to fixation. So that's the setup. That's all you guys need to know in terms of population genetics for the rest of this talk, really. Let me give you some examples now. So the title of the talk is The Tortoise and the Hare. Let me tell you about the tortoise. In 2004, my colleague Gil Bejarano discovered hundreds of sequences in the human genome that were 100% conserved between human, mouse, and rat. Okay? So this was an astounding finding. No one would have expected a priori that there would be huge swaths of the human genome that had been conserved for millions and millions of years of evolution. Even more astounding was the fact that if you went and looked at these so-called ultra-conserved sequences, many of these maintained this conservation all the way to chicken. So just like the comparative anatomy example I was giving you, we're talking about 310 million years of evolution and nearly no change at the level of sequences. Here's an example of what one of these looks like in the genome. If we look at a vertebrate alignment, you can see conservation kind of going up and down here, and here's our ultra-conserved element. Okay, so 100% sequence conservation over millions and millions of years of evolution. Here's the surprising thing from the perspective of a genomicist, right? The gold standard for function in genomes is the gene. Okay, well, where do genes fall in the distribution of sequence conservation? They're somewhere over here. Okay, well, ultra-conserved elements are really out on the tail. So genes, the things that we know do something in the genome, aren't really all that conserved if you look at the whole distribution. Whereas these other elements that we know nothing about, and I'll tell you something about them in a second, are actually the most conserved regions of the genome. So when you look at something like this, there are actually two competing hypotheses, like we've been discussing, to explain the conservation. The first one is the Kimura and Oda hypothesis, that conservation implies function. The second equally plausible hypothesis is that you've stumbled upon a mutational cold spot. They very well could exist. The beautiful thing is now we can use population genetics, population genetic estimation, to untangle these hypotheses. So a little bit of data for you. And this is going to be a kind of data-centric talk for the rest of this. We went and PCR resequenced a bunch of ultra-conserved elements from 96 humans. And for comparison, we're going to look at a set, the Seattle SNPs data set, a set of genes that have been sequenced. And in particular, we're interested in comparing frequencies, the site frequency spectrum, from ultra-conserved regions versus the site frequency spectrum at genes, in particular amino acid changing SNPs, so-called non-synonymous SNPs. So here's the raw data, and we'll have some fun with it in a second. These are the raw site frequency spectra. What I want to point out here is on the right we have our non-synonymous sites. On the left we have ultra-conserved sites. What you can see is that ultra-conserved sites are indeed shifted to the left. 55% have a derived allele frequency of count 1. Okay? There's only one SNP with a frequency greater than 0.5. Whereas non-synonymous sites are shifted to the right with respect to the ultra-conserved elements. Okay? So what does this say about selection? Well, let's do a little modeling and find out. What we're going to do is we're going to fit a Wright-Fisher model with a distribution of selection coefficients. In particular, what we're interested in doing is we're interested in saying, is the distribution of selection coefficients for ultra-conserved elements different from the distribution of selection coefficients at non-synonymous positions of the genome? To do that, we need a simple mathematical model that connects the frequency of alleles to selection. My model here looks at the probability that some SNP is segregating at some frequency xi, conditional on some selection coefficient and some ascertainment. We have to worry about ascertainment, like John was talking about a little bit, and the sample size, of course. Because I'm interested in the distribution of selective effects, I need to fit a slightly more complicated model that we don't really have to get into. But what I'm going to do is I'm going to fit a Bayesian hierarchical model where I make some assumptions. I'm going to assume that there's some unknown genomic distribution of selection coefficients. We're going to assume that that distribution is normal. That is to say that each mutation is going to get its own selection coefficient drawn from some genomic distribution. Okay? We're then going to estimate the mean and the variance of that unknown distribution using MCMC. And I should point out that this is related to the methods of Stan Sawyer and Carlos Bustamante, except I'm allowing for every mutation to have its own selection coefficient. One step removed from that. Formally, this is what the model looks like. Right? We've got something big and ugly down here. This is intractable. We've got our likelihood function, and we've got some priors. So the magic happens. You go and you run your MCMC, and what comes out of it? Well, here's what comes out of it. The mean selection coefficient for our ultra-conserved sites is somewhere over here at -5, whereas for non-synonymous sites, we're at somewhere around -1.6. Okay, and it's worth pointing out that our credible intervals don't even overlap for this set. Okay, so what does this tell us? Well, what this tells us is that, at least from the standpoint of allele frequencies, our most conserved portion of the genome seems to be under very strong selection. In fact, stronger negative selection than our genes, our actual functional elements. Okay? This all screams that these elements are functional. But what are their functions? And what I didn't tell you is that the majority of these elements actually occur in non-coding regions of the human genome. 75% of them are in non-coding regions of the human genome. So what do they do? Well, this is an open and interesting question, and very little progress has been made since these elements were discovered. About 50% of these elements have some kind of enhancer activity. At least a few of them are involved in splicing regulators. Okay, so still we really don't have a handle on what this kind of collection of elements in the genome does. But we really should be chasing that down. Okay, so let's look at the other side of selection. Let's look at the portion of the genome that evolves adaptively. And in particular, I want to ask the question, what are the targets of positive selection in genomes? So what are the logical places to look? Well, the logical places to look might be lineage-specific fixations, like that example I was showing you from FOXP2. And for that, I'll be talking about some hot-off-the-press fly data. And another logical place to look is the rapidly evolving bits of genomes. We know that positive selection hastens the process of fixation. Okay? So rapidly evolving portions of the genome are probably more likely to be under positive selection. So let's look first at adaptive fixations in Drosophila. Drosophila has an interesting demographic history. It mirrors our own. In fact, the peopling, or the peopling from the perspective of flies first got to the New World as a result of people, probably around the time of the actual slave trade. Drosophila melanogaster is thought to have originated in Africa. So the question that I'm interested in asking is, what kind of ecological adaptations accompanied this out-of-Africa migration? That is, when Drosophila melanogaster moved from this tropical environment to this temperate environment, can we pick up the signature of ecology? To do this, I'm going to put things in a phylogenetic framework. So we have a very nice alignment of Drosophila genomes, 12 Drosophila genomes these days. And using the Davis DPGP data, the Drosophila Population Genomics Project data, we now have 37 fully sequenced lines of North American Drosophila melanogaster, as well as a handful of fully sequenced African lines of Drosophila melanogaster. So what I'm interested in doing is I'm interested in identifying fixed differences that have happened along the lineage leading to North America. So if we zoom in on this part of our tree, here we see melanogaster, and we can divide melanogaster substitutions into two classes, those that have occurred throughout the entire population of Drosophila melanogaster, the entire species, and those that have occurred along North America alone. Now, this is the site frequency spectrum version, the perspective from the site frequency spectrum of exactly what John was talking about. So when a selective sweep occurs, okay, selective sweeps reduce variation at linked sites, and they also lead to a skew in the site frequency spectrum at those linked sites. So if we imagine that somewhere on some chromosome, I think here we're on the middle of chromosome 2, there was some selective sweep. As we move away from that selective sweep towards unlinked regions, we can recover our neutral site frequency spectrum, something close to neutral. But as we move in towards our adaptive fixation, if you would, we're going to see a greater and greater perturbation of our site frequency spectrum. This allows us to actually specifically test whether we're looking at a sweep. To do that, what we need is, again, a little probability model. I'm going to elaborate a little bit on a model that Rasmus Nielsen came up with. And I should say that actually John has a hand in this as well, I believe, some of the initial work. But what we're interested in doing is connecting the frequency of a SNP. We're interested in saying what is the probability of observing a SNP with some frequency xi, conditional on a sweep that occurred at some site, and a selection coefficient, and a recombination distance now between our site and the sweep site. This allows us basically to localize sweeps. Once we can write down those probabilities, we can do what comes naturally. We can write down some kind of likelihood ratio. In this case, I've called it a composite likelihood ratio because, as John was pointing out, unfortunately, we don't have full likelihood analytics yet that are available. Instead, what we can do is we can do a pairwise thing. We can say, oh, well, this is the probability of this site, but I can't tell you what the joint probability of all the sites are. So instead, what I'd like to do is give you a true likelihood ratio, but instead of doing that, what we can do is we can approximate that using all of those pairwise comparisons, all of those pairwise probabilities. The hypothesis I'm interested in testing is what is the probability of my data given a sweep in comparison to the probability of my data given no sweep at all. And what I'm going to do is I'm going to look at those individual fixations. Okay? Each of those fixations that I've identified via phylogenetic methods, I can then go and evaluate that. I can do that hypothesis test, if you would. So here's an example of a nice sweep. This is a North America-specific sweep that occurred in a gene involved in eye development, rugose. And what I've plotted here for you, so this is the normal UCSC genome browser. But what you're seeing here is nucleotide diversity decreasing in that nice valley that maybe some of you are familiar with, right around our sweep. Down below this line is actually Tajima's D. So right where our sweep occurred, which is in this exon, what we see is this valley of heterozygosity and this skew in the site frequency spectrum, a very negative Tajima's D for the genome. Okay? That's just an example of what one sweep looks like. So where are the targets of adaptation? Well, these data are brand new, and we're still working through the bugs in this, and there almost assuredly will be bugs. So don't inhale yet. But that said, we can go and divvy up our fixations into those melanogaster-specific fixations and those North American-specific fixations, and what we get is a very different picture. What we see is that along the lineage leading to melanogaster as a species, about 10% of the coding sequence fixations that we observe seem to be adaptive, whereas very few RNA gene fixations are adaptive. We have some evidence of regulatory adaptation taking place. We also surprisingly have some evidence of adaptation in quote-unquote conserved regions. So that's an interesting observation that we haven't squared yet. If we go and look specifically at the North American clade, what do we see? Well, we see this preponderance of RNA gene adaptive fixations. Just to tell you a little story, there's an amazing RNA gene that came up in this screen, something called heat shock RNA omega. okay? An RNA molecule that's known to be involved in cold tolerance, and it's come up numerous times in Ari Hoffman's clinal data from Australia, okay? So everything seems to be working. Oh, and this is something for the aficionados of codon bias, if there are any out there. What you'll see is that just like Hiroshi Akashi and others, including myself, have reported in the past, we're finding adaptive, unpreferred codon fixations happening in North America. It's kind of a wild and consistent result in Drosophila population genetics these days. So, what if you wanted to say something about genes? Well, we can do the kind of natural thing and divvy up genes into functions using the Go categorization. What do we come up with? Well, two very specific processes seem to be coming out. One is courtship, here in red. And the other is eye differentiation between North America and African populations. So I have this up here not really so much to spin stories as much as to give you a tantalizing view as to what a reverse genetic approach to adaptation might be. That is, if you wanted to study adaptation not via crossing things, but truly in a reverse perspective, what would that look like? Okay, so off from flies in a second. Now let's go look at the rapidly evolving portions of the human genome, the hair, my little cartoon down here. What if we want to ask the question, what makes us human? One of the things I like to point out to the students in my introductory class is you don't see this very often, right? You don't see chimpanzees driving cars very often. It's not because it wouldn't behoove them, right? It's probably because there are some genetic differences between us and chimpanzees. So can we go and find the human-specific targets of adaptation? Well, the approach we took in about 2006, 2007 was a phylogenetic approach. This is work that I did with Katie Pollard and Sophie Salama. What we were interested in doing is basically comparing via maximum likelihood phylogenetics two very different models. One in which we allowed for, consider this tree for a second here, where what we're going to allow for is basically no lineage-specific speed-ups. That is, we have some model, okay? There's a fixed rate of evolution along every branch. And we're going to compare the likelihood of that to the likelihood of a tree where we allow for the human branch to have its own rate, okay? And you can do a formal likelihood ratio test and account for the difference in the number of parameters. And of course, the question we're asking is this tree, is this model significantly more probable than this model? And we can get a nice list of hits. We get 50 really good hits. But rather than kind of focus on global patterns, let's look at some of the specifics because there's some interesting stories here. This is our top hit. Let me introduce you to HAR1, Human Accelerated Region 1. This was a non-coding region of the genome that had no known features in it. As we looked a little more closely, what we found is that there was an RNA secondary structure associated with our target. Okay? Sure enough, Sophie was able to go and validate the RNA structure that we had predicted. There were actually two transcripts associated with this region, HAR1R and HAR1F. And amazingly, if you go and you fold the structure of this RNA, what we found is 18 substitutions along the human lineage. It actually makes 9 compensatory substitutions. So that is 9 substitutions where both interacting players were actually substituted. So 18 substitutions from one perspective, 9 from another. There's some evidence that there's a signature of an ancient selective sweep, but I'd like to point out that it's really weak, okay? When you go and you compare the human version, 5 minutes, when you go and you compare the human version of this gene to the chimpanzee version, what do you see? Well, what you see is that in the human version, there are actually new interactions that have been created. Okay, so what about the phenotype? What is this gene involved in? Well, through collaborators over in Holland, we were able to show that HAR1F is specifically expressed in the cerebral cortex. In particular, this gene is co-expressed with a gene called relin, which is involved in laying down the layered structure of the neocortex. Okay, so to me, this is really kind of an amazing confluence, right? we go out and we screen for those regions of the genome that should be involved in human adaptation. What's our number one hit? Well, it's an RNA gene that seems to have something to do with brain development. Okay? It raises the question, does this actually have to do with human brain evolution since our most recent common ancestor with chimpanzees? It also raises the question, well, that was such a successful endeavor. What the heck did these other HARs do? Is selection involved in their evolution? I would say, ask me later about this. Okay, the answer seems to be largely no, which is pretty interesting. But again, like I said, they're such nice stories that it's definitely worth telling. So we got scooped on HAR2. Okay, another group of experimentalists went and characterized this before we could. What they found is that HAR2, here we're looking again at a browser shot. You can see there's a lot of conservation, but you can also see that this region of the genome is littered with human-specific substitutions. Okay? What they found is that the human-specific version of HAR2 seems to drive expression. Let me get below this so I get it right. Seems to drive expression in the forelimb in something analogous to the thumb. Okay, so this is incredible. And what I should point out is that the chimpanzee ortholog does not drive that expression. Okay, just the human-specific version of this. Okay, so something about the human-specific substitutions that have occurred are increasing the reporter gene assay output in just this very specific region. Really an incredible result to me. So listen, those are some examples for you guys. I was asked to give a little bit about the prospectus for evolutionary genomics in general. And while I don't feel very comfortable as a junior member kind of waxing philosophical about where I see the field going, what I will tell you about is what's happened recently. And what's happened recently, overwhelmingly, is that sequencing is moving forward. Genome sequences are going to be available out the wazoo. So this is a slightly complicated slide. What I'm doing here is I'm comparing from 1998, 11 years ago, to 2006, the different sequencing technologies that have come out. On the y-axis, we're looking at the number of base pairs output by one experiment on that instrument or that technique. And down here, I've got the price per base. So back in 1998, in the era of the 377 from ABI, right, you could get about 500 base pairs at a one sequencing reaction for a cost of 1 cent per base. Then we moved up to resequencing arrays. Well, they were pretty expensive, and you got about 300,000 base pairs out of an experiment. In the old 454 technology, you could get about, you were getting close to a megabase of sequence at a cost of about 0.03 cents per base. But in 2006, with the advent of Solexa sequences, you just skyrocketed up, right? Now you're getting a gigabase per experiment at this astronomically tiny price. The Human Genome Project took 13 years and $3 billion. Okay? Now you could do exactly the same thing in about 3 weeks for about $20,000. Absolutely incredible. Okay? This is the slide I like to point out. If that wouldn't be enough for you, if you didn't want to be in the public consortium or something, you could go on to eBay and for $68,000, buy it now, right? You could get your whole genome sequenced. So where do I see the future going? Well, there's no question where the future is going. Everyone's going to have their genome sequenced. Everything is going to have its genome sequenced. This is a treasure trove for people who are interested in evolution. So with that, I'd just like to thank some people. My postdoc at Dartmouth, Brian Kolodjakowski, did all the Drosophila work. He's amazingly talented. You guys should look out for him in your job application pools. At UCSC, I got to thank David Hauser, my postdoctoral advisor, my collaborators Saul, Gil, Katie, and Sophie, and the folks at Davis, Chuck and Dave, who allow me to play with their data. Thanks.